Hello, everybody. We are down to 11 people, which means 50% dropout rate in one class. So next Wednesday, nobody might show up. Okay, let me just quickly go through attendance. Attendance is obviously not mandatory or anything, but I would like to keep a record of who's showing up and when. Yash? Rishi? Naman? Rahul? Akshat?
Yeah.
Devendra?
Yes, sir.
Rishi, this is Rishi Chauhan. The first one was Rishi Dhape. Right. Rohit,
Present, sir.
uh, Shreya,
Yes, sir.
Anirudh,
Present.
Pavitra,
Sir, here.
Namrata,
Yes, sir.
Anju, Sangamitra,
Yes, sir.
Nilesh, Prachita,
Yes, sir.
Sakshi.
Present.
Yes.
Yes, sir.
Okay. Anybody else whose name I didn't call out?
Sir, Akanksha.
Akansha. Yeah. Anybody else?
Yes, Buddhadev.
Are you guys new registrants?
Yes, my registration got confirmed yesterday night only.
Got it. Okay. Anybody else?
Sir,
All right.
Adhishri, I just registered. I
Okay.
also registered.
So, Akanksha, you are a third year undergrad, right?
Yes.
Cool. And Buddhadev and Adhishri, can you just tell me about your academic standing?
I'm a
Uh,
fourth year dual degree student, chemical
In
engineer.
in Chemie, right? Okay.
Yeah.
Okay. Cool.
Sir, I am MTech student of biomedical engineering, first year.
Okay.
I have taken this
Okay.
as institute elective.
Fine. Cool. So this is your second semester at the institute.
Yes, sir.
Okay. Cool. Okay. So a couple of logistics type things. Uh, One of you has already sent me the summary for chapter 4, which is great. We're talking of origin of species, chapter 4 summary, which is great. But this was due on Wednesday. So, I mean, it's great you have done it. And since you have done it, I will go through it and I will send you comments on it. But if you choose to, you can submit again on Wednesday. So before Wednesday's class, 9:30, I would like all of you to submit a summary of chapter 4 of Origin of Species. Has anybody read the Origin of Species before? We've all heard of it, but has anybody read it? Be completely, shameless when it comes to admitting stuff like this. No issues whatsoever. I read it after I became a faculty, so. Okay, so no one. Cool, great. So I will walk you through. I hope time permits at the end of the class today, but I will walk you through the structure of the book, how Darwin lays out his argument. But there are a couple of things that we want to do today before we get to the actual discussion of the book. Okay, so we will pick up from where we left last time around, and just to bring people who are new up to speed, I will sort of start from scratch. Yeah. Let me go. All right. So basically, the tree of life says that we have one species which splits into. Before I start, I of course this online business is a little uncomfortable for everybody. I'm sure it's as comfortable for you guys as it is for me. And of course, the biggest problem I find is that I can't look at faces and tell that whether whatever I'm saying is making sense or not. And it's a huge disadvantage. Uh, so just feel absolutely free to stop me, interrupt me, if I'm not making sense at any point. I mean, again, I think today's lecture is more of a story rather than an actual rigorous discussion. So there is not much that will not make sense, I think. But next time onwards, it is going to get technical. So this will start happening. Unfortunately, I can't see your faces, so just feel free to stop me at any point. And again, please also feel free to stop me for comments, arguments, anything like that. I would like this class to be as dependent on discussions as possible. All right. So what we were discussing last time around when we stopped was that we have species which we have populations which are evolving into more complex entities. And what that explains, the theory of natural selection as we have in our minds, explains that how an existing population in a given environment can get better adapted in that particular environment. But what theory of natural selection doesn't tell us that if we go back in time, what this means what this means is that all of life on Earth must have come from a common ancestor, and that common ancestor has a name. And basically, when we talked about origin of life last time around, this first evidence of life on Earth, roughly 4 billion years ago, the first common ancestor from which all of later diversification of life happened has a name, and it's called Luca, which is last universal common ancestor. What the theory of natural selection doesn't tell us is that how did Luca first come up? And what we ended last class with was a very brief discussion on what all would it take for us to build the simplest possible cell that we can think of. Not a eukaryotic cell with sophisticated compartmentalization of various cellular functions, but a rudimentary a prokaryotic cell that we can think of. And what we sort of listed down as a result of that discussion was that we would need a genetic material, because that could be we're not interested in chemistry at this point, but what we mean is that at some logical level we would need a chemical entity which will contain the information of heredity, because information has to be transferred from one generation to the other, and there has to be some chemical basis via which that information is transferred. It happens to be DNA, but that's a detail that we're not interested in. We definitely need a source of energy, because entropy has a tendency, entropy increases, and living beings are all about fighting against entropy. So we are going against second law of thermodynamics, which means that we have to put in energy to maintain this order within our bodies. If we left it to itself, our body would move towards entropy and decay and not function. So in order to maintain this order in our bodies, we have to input supply energy. And that's true for the first primitive cell as well. We would need catalysts, because much of life is just catalysis. And we would need some sort of catalyst, whether these are enzymes, whether these are protein molecules enzymes, whether these is RNA molecules, whether these are inorganic catalysts that are present in the environment that Luca the last universal common ancestor evolved in is irrelevant, but we would need some sort of catalysis. We would need a membrane because as soon as we are talking about a living entity, that means there is a physical geographical delimitation between what is inside the cell and what is outside the cell. So we will need some sort of a membrane which defines the boundary of an individual. And we would need carbon, nitrogen, assuming it's carbon. I mean, we know it's carbon based because we are all carbon based, but it could be any other element also if we were based on, our life was based on another element. So we need a supply of carbon and other resources like nitrogen, oxygen, phosphorus, sulfur. We would need a supply of all of these elements, and we would need a replication machinery because whatever this information is has to be copied before it's passed on to the next generation. So those are basic constituents that you would need, that we came up with in order for us to have the first most primitive kind of a prokaryotic cell. So the idea of start of life on Earth actually has been around for various ideas have floated around at various times on Earth. In 1870s, Kelvin, Lord Kelvin at that time, argued that the germs of life might have been brought to Earth by some meteorite, which basically transfers the burden of evolution of life from this planet to another planet and still leaves the question unresolved as to how could life have come into being. Lord Kelvin was actually also one of the severe critiques of Darwin's theory of natural selection. And via some calculations that later turned out to be wrong, he estimated that the life of Earth was not very long at all. And thus he argued in the years subsequent to publication of Origin of Species, he argued that Darwin's theory is wrong because Earth hasn't been around for very long. So he was one of the critics. And next Friday, we are going to discuss a critique written by an electrical engineer called Fleming Jenkins. I talked about this briefly last time around. We're going to read Darwin's critique of Darwin's book by Fleming Jenkins next Friday. And Jenkins was actually Kelvin's student. So this notion that origin of life didn't take place on Earth, but was because of just life being transported by a meteorite from another place in the universe, remained a fringe notion until the 60s, when in Australia, a billions of year old meteorite was chemically analyzed and found to contain amino acids, sugars, and nucleotides. So it seemed like the chemistry of life had already taken place billions of years ago at other places in the universe. As against this, the sort of estimates of the estimates of how long life has been around on the planet, and also how long Earth has been around, has been constantly being revisited as we've progressed through the decades. 60s, Earth was thought to be less than a billion years. 70s, it was thought that it was at least 2.5 billion years ago. And our best estimates today are that life is around 4 billion years. And this is not going to move very much now because Earth is around 4.5 billion years old. And the first 300 million years or so, there is consensus that the environment was too volatile, the temperature was too high, there were molten lava flowing on the surface. So really, carbon-based life could not have evolved in that period. So I don't think this number is going to change now very much. So as I said, that this idea has been around for a while, but this merely just transports the question that we want to answer on Earth to another planet in the universe. And it's not a very, to my mind at least, it's not a very satisfactory explanation of what's going on. All right. So Next, I want to discuss Stanley Miller and Leon Urey's experiment. You guys tell me about what you know.
The primitive Earth conditions were mimicked in the experiment by using like lightning source.
Okay. Others?
There was some kind of so they added the basic elements required for synthesis of any amino acid or anything, and they actually found amino acid production, I think, through the experiment. So that's how they concluded that probably this was the way it started.
Yeah, right. So again, I remember this is early 50s. So not a whole lot is known about what are the information molecules, which molecule is responsible for what particular role inside the cell, what are the molecules of heredity, but what is known is that proteins are extremely important, and proteins are catalysts, and proteins are chains of amino acids. So really, the biggest puzzle that needs to be resolved at this point, around 1950, is explaining how did proteins come into being. And explaining proteins comes into being seems like a quiz, seems like a puzzle which requires you to answer where did amino acids come into being. So Miller, who was a student in Urey's lab, came up with this experiment. But the idea of the experiment had been around for a long time. Popperin and Haldane called it the primordial soup. There was consensus that life needed water as a solvent to have started from. It couldn't have taken place on land. So the primordial soup idea was there in Popperin and Haldane. Darwin wrote a book in 1871, The Descent of Man. And in this one, he talks about how life could have originated on Earth. And he writes that, but if, and oh, what a big if, we could conceive in some small, warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity present, that a protein compound was chemically formed, ready to undergo still more complex changes. So this seems like a pretty big hint as to how can we go about doing this experiment. And it was present to undergo still more complex changes. Basically means that he's envisioning that Darwin didn't know proteins at the time, but in the 50s now we're thinking that, oh, if we can get proteins made, and we know that proteins have catalytic functions, and if we can get these catalysts formed, that's a great start towards explaining how greater complexity could have arisen from starting with just what Popperin and Haldane called a primordial soup. So this was published in 1953 in Science. And the experiment is essentially, as you guys have read about in school, I certainly read about this in this when I was a school student. And essentially what's happening is that there is water, and this just mimics the fact that there are oceans. The air above this water pool is reducing in nature because there was hardly any free oxygen present in primitive atmosphere. When you're passing through sparks, low voltage electricity, and there is a heat source to keep the temperature around the levels of 40 to 50 degrees. And what you're starting, and in the water, of course, you added some phosphorus and nitrogen and sulfur salts. And what Miller saw is that after running this experiment for as short a period as a couple of days, the liquid, which was clear to begin with, turned pink. And after a week or so, the liquid became brown in color. So something was clearly happening chemically. And when he analyzed using the analytical techniques of the time, he found that uh, there were 8 amino acids present in the liquid, in the brown liquid that had been formed by that had been transformed via chemical reactions that are taking place. Are there people who do not know what are amino acids chemically? We can just take a quick look. Raise your hands, please. Everybody is aware what is chemically what is an amino acid?
Yes.
Okay. How many different types of amino acids combine to form proteins in living entities?
22.
Others? Guys, I do need help from you regarding whether we know this part or we do not know this part. No help? So this is what an amino acid would look like. So essentially there is a carbon, and this carbon is attached to a hydrogen, a COOH group, a NH2 group, and an R group. And this R group could be anything. And the proteins that we have in our bodies, we use 20 different amino acids where and each amino acid is different from the other because of the different R that's present. This is fairly straightforward. So everything else remains the same. So you have the next amino acid, which will link with this current amino acid. And basically this NH2 of the next amino acid will combine with the COOH of this amino acid, and you will have a dehydration reaction take place, and a chain of amino acids is formed, and that constitutes the unfolded protein, and then it folds into its 3D structure and does its job inside the cell. So what Miller found that after a week or so, when he analyzed the samples that he was looking at, he found 8 different amino acids present in the liquid pond. So amino acids with 8 distinct R's. And commenting on, and this was published in 1953 in Science. And as I said, not yesterday, last time around, this was a fairly big deal. In fact, at the end of 1953, Stanley Miller was on the cover of the Time magazine. This paper was thought to be much bigger than the Watson and Crick paper, which published the structure of DNA, also in Science. So, and Urey, commenting on the paper, said that, and God is in quotes, I guess here, if God didn't do it this way, he missed a good bet, because this seemed like an excellent way of how life could have originated. Miller passed away in 2007. And interestingly enough, the samples from the 1953 experiment, he had preserved little volumes of the samples from that experiment. And many of his experiments many of his students sort of revived the preserved samples from over 50 years back, and using the much more sophisticated analytical techniques, analyze those samples again. And in 2008, they published a paper in PNAS where they actually found out that Miller's primordial soup did not have just 8 amino acids. It had 23 amino acids. 15 of them were just not detected because of the lack of precision or sensitivity of the analytical techniques used at the time. And after publication of this paper, this was repeated in 1,000 different ways. Somebody would say that no, methane should be slightly more, ammonia should be less in the atmospheric atmospheric air composition that you're trying to mimic in the experiment. People tried different chemistries of salts that were present in the liquid pool. And irrespective of whatever concentrations and amounts of the constituents in liquid and the air that you used in this experiment, you would always get amino acids. So this seemed like a very, very robust experiment and a great way to make amino acids. And it seemed like once you have these constituents for proteins floating around freely in ocean that you're trying to mimic here, from there on, it just seemed like a matter of time before you could come up with a way to explain how, resulting from these amino acids, you could have proteins form. And that would be a great start.
Yeah, I have a question here.
Yeah.
So when he mentioned 23 amino acids, are they the 23 general 20? I mean, are they part of the 20 general amino acids, or are they groups very different from the general 20 amino acids?
No, so I forget the exact number, but I think at least 11 or 12 are the ones which sort of constitute proteins. But because it's greater than 20, definitely there are at least a few which are not part of protein or the genetic code. But still, I mean, this is just chemistry taking place, right? This is not origin of life. Yeah.
Okay, thanks.
So yeah, I can share that 2008 reference. I forget the exact number, but I think it's 12 or 13 which are part of proteins, and the remaining 10 or 11 are the ones which are just chemicals, which are not part of life. All right. Okay. So interestingly enough, this obviously was an important and a big paper, but when this was published, Stanley Miller was the only author of this paper. Stanley Miller, remember, was the young graduate student in Urey's lab. Can you imagine why the advisor would not put his name? I want you to guess why would that be so. If you know the answer, keep quiet. So this was a paper in Science, an important paper in Science. Why would the advisor's name not be there on the list of authors? Miller was the only author of this paper. Go for it. Anything.
Maybe because it went against the conventional thought at that time.
Right. So what you're saying is that maybe Urey intellectually disagreed with it.
Yeah, maybe he didn't approve of what he said.
Yeah. Sure, but I mean, in terms of results, you can't challenge the results because it's right there. It's happening right in front of you, right? So you would need a strong reason to. So you can't doubt the results. Experiment done this way does lead to formation of amino acids. That's just chemistry happening right in front of your eyes. And it's not even a very complex or a long experiment to repeat. You do it for 2 days and the water turns colored. So there's clearly something going on. So you can't doubt the chemistry. So if it was a disagreement,
Was it religious?
What was that?
Was he very religious and it didn't
Interesting
come into
idea.
account?
Yeah, I don't know if Urey was religious, but I would say that if it was my lab and I believed in divine creation, then I wouldn't let an experiment like this take place in my lab, right? It wouldn't come to a stage where it's come to publication and I would withdraw my name. I probably wouldn't let an experiment like that take place in my lab. So that's not the reason, but both of you, good guesses. Other ideas?
Was Ure banned by the scientific society for some reason?
No, no, he wasn't. No, he wasn't. And I laugh because of a particular reason, which I'll tell you when I share the answer. So the reason why Ure didn't want his name put on the author list was because he felt that if his name was there, Miller wouldn't get credit for the paper. People would think it was the advisor's idea, and the student wouldn't get the credit for it, which is as generous a story in science as I have ever heard. Uh, does that surprise you guys?
Yes, it does.
Yeah, it shouldn't because of what's coming next. Because Urey only did it because he had already won a Nobel Prize 20 years back. Okay, so he didn't really care for a science paper. You know the hydrogen isotope deuterium?
Yeah.
Yes.
Urey discovered that. So he had already won a Nobel Prize for that, and he really didn't care for another science paper. All right. So back to this. Now, this experiment sort of this was very much part of textbooks about origin of life when I was I don't remember, but maybe a 8th grade student or something like this. This was a big part of our textbooks. Can you think of a problem with this idea? Not the chemistry. We can't doubt the chemistry because, as I said, in the decades that followed the publication of this paper, numerous variations of this experiment were done, and it would lead to amino acids being detected in the mixture in a very short space of time, very, very soon. What could be the problems with this experiment in terms of being able to explain the origin of life, which was the premise of this experiment? Remember, the premise wasn't to show that nucleotides or amino acids form. The premise was the premise is to create conditions which could explain origin of life. Any ideas?
Sir,
Yeah.
this only shows the origin of amino acids and not life as such.
Right.
So it
So
might take more for life to form.
correct. But again, exactly. But I want you to sharpen your logic here, sharpen your critique here. So what would it take? So you have this experiment has, I think, very convincingly, this and the subsequent experiments by other people have very convincingly exhibited that formation of amino acids happens really fast. Now, I think in an experimental setting, you probably don't expect that in a pool like this, cells will form, right? That's too much to expect, right? The next step in this experiment would be explaining how these amino acids would come together to form an enzyme which has catalytic functions, which would mean that we have to explain formation of protein molecules from these amino acid, from this pool of amino acids that's been formed. And now we have to expand this experiment into the ocean. And can you think of a problem that might exist in terms of this experiment being able to explain how enzyme molecules could start to appear from this pool of amino acids that's been generated? Is the question clear?
Can you repeat?
So the idea is the larger context of this experiment is proposing a mechanism by which life originated. And by life originated, we mean cells came into being. The most primitive kind of cells came into being. Now, in an experiment like this, which runs for a week, a month, even a year, 10 years, we can't expect cells to show up in a pool like this, right? That's unrealistic. But what would be a great step towards explaining evolution of life would be, and the fact that amino acids appear cannot be contested because they appear very rapidly and repeated throughout the world. But what would be a crucial step towards moving from appearance of amino acids to appearance of life would be these amino acids come together to form protein molecules, right? In free ocean, do you think that will happen? Now let's forget the experiment and let's imagine that we have amino acids in primitive ocean freely floating. Now they have to come together to form enzyme molecules. Do you think that would happen? And if not, why that would not happen?
Like today we have machinery that helps in the synthesis of proteins. But in a primitive pool, it may happen, but that would be very disorganized.
What do you mean disorganized?
There might be oxidations or reductions.
In oxidation,
Random
we don't
reactions.
have to worry about too much. We don't have to worry about oxidation too much because there is no free oxygen. Trace amounts of oxygen is present. But what would it take for these two amino acids to come together and form a peptide chain?
There should be a lot of amino acids, and exactly the amino acids that form the protein should find each other and collide and then form a bond.
Yeah, the first critique is a valid one, that there should be a lot of amino acids, right? In a free ocean, it's really hard to imagine that state being met, that condition being met. The second one, we don't have to worry about too much because we don't care which protein gets formed. All sorts of combinations will get formed, but if a catalytic amino acid chain is formed, that serves our purpose, and the rest can degrade or whatever might happen. We're not worried about that bit. But the main critique, let's return to this, the main critique of this idea, which started in the late 80s, was the fact that in free ocean, if you did have amino acids present in the ocean pool, two things need to happen. These amino acids need to come together and a dehydration reaction needs to take place in the ocean where their water molecule is stripped away and you get the peptide chain. This needs to happen without the presence of any catalyst. Now in free ocean, in extremely small amounts of amino acid concentrations that might be present there, this is a really hard step to imagine. So the first problem is that of concentration. The second problem, also associated with the same thing, is the fact that concentrations are small, but the dehydration reaction requires energy. And in free ocean, what is that source of energy? That source of energy is going to come from the sun. And there have been some back of the envelope calculations which requires that events highly improbable events like lightning need to happen at the same spot in the ocean, again an exceedingly improbable number of times for enough energy to be there for that dehydration reaction to take place. And as a result, the opposition to this idea started gaining strength. Something changed then. So you guys know, okay, let me explain this. It's a really curious thing that all living beings make. Do you guys know what is ATP?
Yes.
Yes.
The energy currency.
Yeah. Others, those who are not from biology background?
Adenosine triphosphate.
Yes, that is its name. But
Yes, like it gets converted to ADP, releasing the energy. Like
correct.
that's what.
Correct.
The pyrophosphate, the bond contains energy.
Yeah, so it's a high energy molecule. It has a triphosphate bond present in it. And all living entities use this as the molecule in which energy is stored. And whenever energy is required for any intracellular cell function, the ATP is converted to ADP with the release of an inorganic phosphate, and energy is released. So it's a molecule in which energy is stored inside the cell. In fact, one of the major measures of how well a particular cell is doing energetically is the ratio of the number of ATP molecules to the ratio of ADP molecules that are present inside a cell. So you grow cells in a particular environment and you measure the ATP to ADP ratio, and you get a fairly good estimate of what is the energetic state of the cell in that particular condition. So ATP is made in a very interesting manner in all cells, and the mechanism has to do with the following. When we eat food, what does that food do?
Breaks down into simpler substances and then energy.
Yeah. So basically, we are eating reduced forms of carbon, and this carbon that gets oxidized to carbon dioxide if full oxidation is taking place. And when those, so it's a redox reaction. There is a oxidation taking place of the food, and in turn something else needs to get reduced with it also. What needs to happen is that when this is what happens, let's say, in our cells, that the food that we eat supplies electrons, and those electrons, and this is the cellular membrane, the gray bar, this is outside of the cell, this is inside of the cell. Forget about the chemistry, it doesn't really matter. The simple logic of it is this: the food that we eat supplies electrons because it's getting oxidized. These electrons are passed through a series of proteins which are embedded in the membrane, right? And because this series of proteins, because it transfers electrons from one protein to another, is called electron transport chain, ETC, right? The name doesn't matter. While it's transferring electrons from one to another, what happens is that protons get pumped out from inside of the cell to the outside of the cell. That is the main job of this ETC, to transport electrons so that it's able to use the potential of the, so these electrons are going from high potential to low potential, and as this energy is reduced here, that energy is used to pump out protons from the cell. And eventually these electrons need to be dumped somewhere, and that eventual dumping station is oxygen in our cases, which gets reduced to H2O or CO2, and that's the end result of complete oxidation of our food. We eat food because we want to pump out protons. Once these protons are pumped out, what you have is a proton gradient across the membrane. The inside pH is higher, the outside pH is smaller. What then happens, and this happens in all living beings, is that these protons now want to come down from this high concentration to low concentration region, and they do that while rushing through another machine in the membrane of the cell, which is called ATP synthase. Called that because it helps synthesize ATP. As these protons rush down this protein, it's like a windmill sort of that gets rotated by the flow of protons that takes place through this machine, and that energy is used to convert ADP to ATP by the addition of an inorganic phosphate here, and hence ATP is generated, right? At the time that this was discovered, this was thought to be an extremely non-intuitive way of making chemicals. Prior to that, in most of 1950s, people were trying to explain how is ATP formed inside a cell, and most explanations, people didn't know how ATP can form inside a cell, and most people believed that the way it would happen is that you would have ADP which would come in contact with another molecule X, and this reaction would lead to X changing into Y and ADP changing into ATP. But this would have to be accomplished by a physical contact of these two molecules, ADP and X. So there was this hunt going on for this molecule X, and a series of papers were written on this subject. What Peter Mitchell wrote in a paper, he found out in his experiments that you could never explain appearance of ATP in an in vitro experiment, in an outside the cell experiment, where you did not have membranes. So it seemed like membranes had a key role to play when synthesis of ATP had to be explained, and which was extremely unusual. And this mechanistic basis of production of ATP inside the cell was proposed by Peter Mitchell. This is called oxidative phosphorylation. And then what happened, and again, it's a great scientific story in a sense, that when this was published, there was brutal attack on Peter Mitchell, because the entire world was looking for this molecule X which combines with ADP to lead to production of ATP. And here's Peter Mitchell who says that no, it has nothing, there is no X. It's just the membranes, protons flow through it, and ATP is produced. So this is like a radical departure from the conventional thought. And as radical departures go, he won a Nobel Prize on this, a Nobel Prize for this, for his troubles. And there were considerable troubles when there's this period from the publication of the paper to the mid-70s, when it became widely accepted that Peter Mitchell was right. That period is called OXPHOS wars, oxidative phosphorylation wars, where he was brutally attacked to the extent that he had to leave his university position because the atmosphere was so toxic against him, and sort of retire into his country house just to recuperate from the toll that it was taking on him. What does this story have to do with origin of life and the problems with Stanley Miller's experiment? Now, again, I want you to guess, if all life form uses the same mechanism to accomplish something, in this case, production of ATP, if all life forms uses the same mechanism, what can we say about that mechanism? Well, let me go a step back. Do you think this is a non-intuitive way of making ATP?
Yeah.
Right. It seems like the intuitive thing would be what everybody else was thinking and search for molecule X, but nobody found it. And this seemed to be a non-intuitive way of making ATP. So the second question now, if everybody is making ATP the same way, what does it mean? In terms of an evolutionary thought, if every organism, every species is making ATP the same way, what does it mean?
That it's the most
Essential
efficient
for
way.
One at a time, Sangamitra.
Essential for survival.
It's essential for survival, but we're looking for something more than that.
It's the most efficient way.
As we will see in the course, that's a really difficult claim to make and impossible to prove.
It originated from the same thing.
Absolutely, exactly. So now, if everybody is using the same mechanism to produce ATP, then that can be explained via one of two reasons. The first reason is that somehow every species stumbled on the same way of making energy, right? The second explanation is that Luca made energy the same way and everybody just inherited the same way. Which one do you think is more likely?
The second one.
The second one, right? If everybody is doing the
Yes.
same thing, it's really impossible that everybody arrived at the same exact solution independent of everybody else. That's too improbable. It probably means that Luca did it this way. So if that's the case, that was the clue that people needed to depart from Miller's explanation. This Oxford Wars, I mean, by mid 70s, it was widely accepted that this was the way ATP was made, and hence the Nobel Prize 3 years from 75. We'll skip this slide, and the departure that has taken place has been in the last 10-15 years or so. And that departure started in 1977 by detecting structures at the bottom of the ocean, which are called black smokers. Black smokers. So what you have to imagine, let me visually show this to you using my hands. So what people saw in 77 were these structures like black smokers. To understand what are black smokers, imagine this. This is the surface of the ocean. Above this is air. This is the bed of the ocean. We're not concerned with surface of the ocean, so this we forget about it. This is the bed of the ocean. There is water here. Then there is rock, and then there is molten metals underneath. Then there's a several kilometers gap here. At several places in the bed of the ocean, there are fissures in the bed, and water seeps down. And as water seeps down, the pressure increases and temperature increases. And at some places, the cracks are so great that they reach the molten metals. And when they reach the molten metals, by the time water reaches there, the pressure and temperature is extremely high. At that high temperature and pressure, when this water comes in contact with molten metal, there's a violent reaction. And then basically there is sort of like a mini eruption, which brings up these metals, the rock that's in the fissure, water vapor, and all of this rushes out. And when this rushes out, it comes out of the ocean bed in a violent way. And as soon as it comes in contact with the cold liquid that's surrounding it, it solidifies into these structures, which are porous in nature, and both gases and liquids are flowing through it. Does that make some intuitive sense?
Yes.
Yeah.
Yes.
So these structures are called black smokers because of the reason that there is black smoke that comes out of them, right? These structures were discovered in 1977. In the year 2000, in a submarine expedition for completely other purposes, somebody called Deborah Kelly saw a different variant of these black smokers. The problem with the black smokers is that when the gases come out and these structures are formed, the structures are often very short-lived, a few decades at most, and the gases coming out are still at very high temperature, not best suited for carbon-based life's evolution. But in 2000, slightly milder version of these structures were discovered by Deborah Kelly, and these are called alkaline hydrothermal vents. What happens in these vents is that when water is passing through the bed of the ocean, it doesn't reach the molten liquids which are floating underneath the bed. It only goes, the crack is not so deep, but the temperature pressure is high enough that reactions take place and there is a mini eruption that takes place. And again, rock and some solid metal embedded in the rock, all of that comes out, vapor comes out, gases come out, and all of this comes. And again, when this comes out of the bed, the cold liquid that surrounds the bed of the ocean, it quickly solidifies into these extremely porous structure. I mean, those of you who've seen how catalysts look like, extremely porous, high surface area, it settles into these. It settles into and makes these porous structures, which are called alkaline hydrothermal vents. They're alkaline because the gases that are coming out are alkaline in nature, right? So and in primitive Earth, because carbon dioxide was present in abundance in the atmosphere, and carbon dioxide is obviously acidic. It's thought that the carbon dioxide concentration was of the order of 1,000 times what it's now. So ocean was acidic, and you have these hydrothermal vents that were formed, which had basic gases coming out of them. So this is a picture of Deborah Kelly's discovery of these alkaline hydrothermal vents. And these vents, these alkaline hydrothermal vents, compared to the black smokers, live for a very long time. So these are zoomed in pictures. You can see pores and stuff. So there is a lot of space, a lot of surface area available for catalysis to take place. Inside of this structures would take place. Because this is porous, you would have these small channels where inside is basic because of the alkaline gases coming through. Outside is acidic because of the ocean being acidic. So you have this natural pH gradient that's formed through this. For this, and additionally, what you have is that you have a lot of iron sulfur and nickel sulfur clusters which are present on the walls of these structures. And these are thought to be important because in the absence of organic catalysts, enzymes, you would need some sort of inorganic catalysts to drive home the earliest reactions that would take place towards evolution of life. And these iron sulfur clusters, nickel sulfur clusters are known to be embedded in these walls of this highly porous structure, which would perhaps be a catalyst before enzymes evolve. I don't want to get into too much detail of this, but essentially for this and many other reasons, the currently held thought is that it's very possible that the first life arose in these alkaline hydrothermal vents. This is an experiment that can never be proven, but even if we were to make an alkaline hydrothermal vent in a lab and do this reaction for 10,000 years, and even if it led to formation of a cell, we still wouldn't know if that is indeed the way it happened on Earth 4 billion years ago, because we just never know whether if the same event happened or not all those years ago. But today, this is the best candidate that we have, which goes towards explaining evolution of life on Earth. So can this be tested in lab? This is being tested in lab, in one lab that I know of. This is in University College London, in a person called Nick Lane's lab. And what he's done, he's basically doing Miller's experiment with these alkaline hydrothermal vents. Since 2000, these vents have been studied in much greater detail now, and we know the exact chemistry that comprises these vents. We know the exact chemistry that is going underneath, which leads to the gases coming out from these vents. And we have an estimate of what did primitive oceans look like. So what Nick Lane is doing is he's created these alkaline hydrothermal vents in lab, and via an injector at the bottom, so this is primitive ocean, via an injector, alkaline gases constituting everything that underlying reaction here would constitute are being pumped through this. And then you're looking for what happens in this. Again, I will share references. This experiment is extremely rapid, extremely new. And I know of only 2 or 3 publications that have come out of this so far. I mean, it's not even been 10 years since this experiment was started. This is something that I think is really exciting that's happened in the last 10, 15 years, which has changed our perception of how life and where life originated on the planet. If you're interested in more details about this, Nick Lane is also a terrific science communication writer, and his books are not very technically dense, but they are not trivial either. So I think he strikes just the right balance for people with your background, and I certainly enjoyed it a lot when I read it about 10 years back. And the book is called The Vital Question. And the vital question, of course, is where and how did life originate on Earth? Just before I end, I've just listed these things down because it's not as if this is the only, I don't want to give the impression that this is the only contender today which is being thought of as the place and mechanism of how life came into being. There are at least two others. Again, I've given references here if you want to browse. These are review articles, fairly straightforward, but there are problems associated with both these ideas. The proponent of first one is David Deamer at UCSC, and the other one is a guy at Cambridge, John Sutherland. Sutherland says that actually a lot of this is a good paper, JME paper in 2015, if you want to skim through it. And basically what Deamer is trying to explain is that how did polymers form from Miller's experiment? So the problem for Miller's experiment is explaining how did polymers from the monomeric amino acids that were formed. And Deamer is trying to solve that problem via a completely different mechanism. So he is saying that it did happen the way Miller said, and in order to overcome the objections of how could polymerization of amino acids take place in free ocean, he's going a different way, which I think is plausible. The other big idea is by the main proponent is Sutherland at Cambridge, who says that life originated by using cyanide as a carbon source. And he focuses on cyanide, excuse me, and he focuses on cyanide because if you are deriving carbon from cyanide for life, the chemistry becomes extremely easy and favorable. The thermodynamics, kinetics, all becomes easy. The problem with this idea is that if we are saying that life arose by using carbon from cyanide, then every single life form that we see around us had to unlearn that had to unlearn using cyanide as a carbon source. And that seems like, and for almost every life form that we know, cyanide is toxic now. So what the challenge before Sutherland is to explaining that, okay, in primitive Earth, cyanide was the preferred source of carbon because chemistry was favorable and there were no catalysts, so that was a big help. But eventually when life diversified, not only did they stop using that cyanide as a carbon source, everyone did it, and also cyanide became toxic for everyone, which couldn't have been the case when life arose because they did use cyanide for life. So challenges remain for every single one of these theories. But as of now, alkaline hydrothermal vents is the most popular notion that we have around. And who is to say, just as Miller's experiment was thought to be right, or thought to be on the right track for 30 years or so, there's absolutely no guarantee that 30 years from now, we'll be saying the same thing about Lane's experiment about the alkaline hydrothermal vents. Okay, so that completes this part. Any questions with this? I still do want to cover something else before we end for today.
Sir?
Yes.
Sir, even fauna is, with cyanide is poisonous for fauna as well?
Yes.
Okay.
Okay. No other questions? So I want to, we have 20 minutes and I will be a little quick, but I do want to discuss the context in which Origin of Species was written. And again, a reminder that chapter 4 summary is due before 9:30 on Wednesday. All right. So I think this is one of the greatest scientific stories that certainly I know of. We have three characters in our stories: Charles Darwin, Alfred Russel Wallace, and Henry Bates. How many of you have heard of Wallace?
Yes. Heard.
Others? How many of you have not heard of Wallace? Please raise your hands. Not physical ones. Everybody else says, okay, see more hands go up. All right, cool. Okay, so our story has three characters. I will be brief. We will end this we only have 20 minutes now.
So, is it Wallace and Darwin, like,
Yes.
did they co-write it together, natural selection?
No, they did not co-write it. They wrote it independently, and why and how that event happened is the story I want to share now.
Okay.
They weren't co-authors. They wrote it independently. All right. So the third guy is Henry Bates. Okay. So here is the story: that Bates and Wallace, not Darwin, are in South America to collect samples. And 19th century science was such that if you were a rich person, that's when you did science. Science really wasn't an employment generation. All the scientists of the time, naturalists of the time, were people who were endowed with a lot of money. Wallace was an exception. He got into science because of his interest, and he wanted to. The idea for him was to just collect samples from South America and send it back to England for private collectors and museums and make money out of those sales and fund his next scientific expedition. The story goes that these two guys were working together. They eventually fell out of each other, went their separate ways in South America. While Bates remained there, Wallace returned to England in 19. Oops, this obviously should be 1852. Wallace returned to England in 1852. And while they're there, the question that they're interested in: where do species come from? Now, so Wallace is returning, and as he's coming back, his ship catches fire. And all the specimens that he's bringing back to England, his notes, everything gets burnt down, and he survives in a lifeboat. And he comes back to England taking a vow that he will never go sailing again. I mean, he's practically been ruined. He's lived several days on a lifeboat and was rescued by a ship passing by. Bates is still in South America, and we'll come back to these two guys. They do not know that Darwin, when he took his Beagle journey, so Darwin was 22 when he started the Beagle journey from Plymouth here. His journey spent a lot of time. The mandate of the ship, HMS Beagle, was to map out coordinates of South America and various other parts of the world. So the ship spent several years, the first, I think at least 3 years, on the east coast of South America, just mapping coordinates. The ship would just keep going up and down. And Darwin would just leave the ship at one place and just go inland for several months and join the ship at another place. The ship goes round, and this is where the famous Galapagos Islands are, off the coast of Ecuador. This is where Darwin saw finches and turtles that form the famous story. And Darwin is seasick throughout this journey. He's not a good traveler, but he also knows that he's on something extremely important that would be pivotal in how the world views species. So he sticks with it because he also knows that he's not going to get another chance like this. He only got approval of his father because of intervention of several family members who then let him go. The ship returns. Darwin's very eager to come back home, but the captain, Fitzroy, decides that, oh, there is one coordinate that we didn't do very well in South America, so we're going to take a detour and go to South America again before making way back home. So he gets back in 1836, and he's tied up in a lot of projects, and he spent several years working on barnacles in the late 40s. immediate years after coming back to England, he spent on just classifying and talking about the various life forms that he saw on this journey. So his first book that he published after coming back was called Journey on HMS Beagle. And that established Darwin as a very famous naturalist of England, one of the leading naturalists that lived in England. Privately, he started keeping a series of notebooks which are called transmutation notebooks. And privately, he started jotting down things about where he thought that species came from. Before this time, the primary view was that species are immutable and they have been created in the present shape that we see them in and have been placed on Earth like this. And they have always existed like this and they do not change. But in this series of secret transmutation notebooks, this is a very famous picture from one of, I think this is transmutation notebook B. He thinks that this is what's happening, that this was a common ancestor which split into different species, and then this one split into different species. So there is diversification of life where these one ancestral species splits into two or more species, and so on and so forth. And this is radial expansion going on. So starting from this guy, you have so many species now. So he challenges the view that species are immutable. And this is a very famous picture from one of his notebooks. But these are his private views. No one knows about them. His public view on evolution is very guarded. In the book that I just mentioned, the first book he wrote after coming back, The Voyage of Beagle, on evolution, he says that it's clear that several islands, and he's talking of Galapagos Islands, have each their peculiar species of the same genre. When these are placed together, they will have a wide range of character. But there is not enough space in this work to enter on this curious subject. So he completely sidesteps the issue of evolution, doesn't want to talk about it here. And Wallace reads this book and thinks it's an open question. And this was one of the reasons which prompted this journey by Wallace and Bates. Why do you think Darwin sidesteps the issue of evolution in this book?
He was gathering more evidence, like theoretical evidence.
Correct. Other reasons?
He's probably afraid of the reaction he might get.
Also correct. Others? Yeah, so no, good. So Darwin didn't really do experiments himself. I mean, not in an exhaustive way anyway. But he did correspond heavily with breeders, horse breeders, pigeon breeders, florists, and he wanted to understand how do they breed certain pigeons so a newer species is born, is come into being. And when all these guys relied on their intuition about these things, and he wanted to understand that intuition. So he was communicating with them, and he was very patient about publishing his views on evolution. And he was also concerned about what Anirudh said, the backlash that he's going to get from the clergy of the time. In addition, he got married in 1839 to one of his cousins, Emma, and Emma was deeply religious and believed in divine creation. And she later on in the 40s came to know of his views on the mutability of species, and she feared for him. And he was careful not to offend Emma in any way by his scientific views. So in fact, eventually when he publishes Origin of Species, he does not talk about evolution of human beings at all. So, I mean, there are also several social and cultural and political reasons to why he does not talk about it so soon. But primarily, the main scientific reason is that he knows that there is going to be a backlash. When he does talk about evolution, he wants it to be a comprehensive document with everything that he knows on the subject. And he he thinks that it's going to be a magnus opus, a huge book with so much evidence that nobody can refute it. And he knows it's going to take time. So doesn't want to talk about it now. On the other hand, Wallace, who had just the previous year taken a vow never to go sailing again, goes back to sea the following year. This time he goes to the Malay Peninsula, primarily Indonesia now. And basically, he did the same thing which Darwin did in Galapagos Islands, but in 1853. And he saw what Darwin saw with his finches and tortoise in Galapagos. Wallace saw with butterflies, marsupials, and monkeys. And a theory begins to take shape in Wallace's mind. And around the same time, both men, Darwin read it much before Wallace, but a heavy influence on both the men was this little book which was published by Thomas Malthus at the end of 18th century, called An Essay on the Principle of Population. And what Malthus was an economist, and what he's saying is that food production for human populations is increasing as we bring more area under agriculture. So food production is linear. If you increase the area by double, the food production will increase by a factor of 2. But human population, if we model it, is increasing exponentially. And exponential growth is going to outstrip linear growth at some point in time. And from there on, once demand has outgrown supply, there is going to be competition. And this competition is going to lead to a struggle for survival. Both men read this as essay and are heavily influenced by it. As a result of this, reading this essay, before we get into Wallace's write-up on this, Darwin wrote a short condensed version of his views on evolution in 1842, and he doesn't show it to anybody. In 1844, he expands it to about 200 pages, still doesn't show it to anybody, but he does keep it locked in his drawer. And Darwin was physically a sick man throughout his life. He tried all sorts of therapies to cure himself of his sicknesses. And in 1844, when he was particularly unwell, he wasn't sure if he was going to make it. And when he wrote the 200-page draft of Origin of Species, he knew it was an important document that could have profound implications. So he wrote a note for his wife and said that in case I were not to make it, please make sure this gets published because this could be potentially important. The two people that he does confide in are a geologist and his mentor, Charles Lyell, and a botanist and a collaborator, Joseph Hooker. And these characters enter our story in a couple of slides. In the meantime, Wallace has read this, and Wallace writes a paper in 1855 about a law which explains introduction of new species. And in this paper in 55, he writes that every species has come into existence coincident both in space and time with a pre-existing closely allied species. What Wallace is saying that there is one existing species, and this one existing species in a geographical location, and something would happen to this species in the prevailing environment where this would split into two closely related species. So there is coincident, these two closely related species are coincident in time, in that they appear at the same time, and also in space, that they appear in the same space where there was originally only one species. And the best mode of representing this natural arrangement of species is a branching tree. This is exactly what the Transmutation Notebook Darwin was writing 18 years ago. And when he sent his paper to Europe, the reaction in Europe, again, he was funded by private collectors and museums to go to Europe and collect these samples and send them, and in return, he was paid. And when he sent this paper back, the reaction in Europe was that he better just keep focusing on collections and not get too much into scientific theorizing. Wallace then, in 1858, writes to Darwin, who is a well-known naturalist at the time, And he writes a letter along with a note that he's prepared for Darwin and says that, will you please read this note and make this accessible to Charles Lyell, who is a very famous geologist at the time. In the meantime, Darwin is writing Origin of Species, which he considers to be a magnus opus which is going to run into thousands of pages. The language of the note that Wallace sends to Darwin, let's just compare a few things. So he says, the life of wild animals is a struggle for existence and to provide for an infant offspring. Whereas Darwin in chapter 5, which is about the laws of variation, says that struggle for existence as bearing on natural selection. All nature is at war. The struggle often falls on the egg and the seed and on the seedling. Exactly the same thing. Wallace goes on, perhaps all of variation must have some definite effect, however slight in the habits and capacities of the individual. A variety having slightly increased powers must inevitably in time acquire superiority in numbers. That means if you have a favorable variation in a population, however small your advantage might be, if you are 1% fitter than everybody else in the population, that means you will, in time, you will take over the entire population. Darwin says any variation, however infinitely slight, if it did promote during any part of life, even in the slightest degree, the welfare of the beings, such variation would tend to be preserved or selected. And the language of the two guys is the same. Of course, Wallace sends it to Darwin. So now imagine when Darwin receives this letter, and this letter says that I've hit upon a theory which could explain origin of species. Could you please read this? Let me know your thoughts and also share this with Charles Lyell. What would be Darwin's reaction?
He might have been confident about his own work now.
Well, that's one way of looking at it. Others?
He'll publish whatever he has as soon as possible.
Absolutely.
Yeah.
Yeah, you would be scared now, because here you are thinking that you've been sitting on a great idea for 20 years, and suddenly some out of the blue comes this letter which might scoop you. I mean, this in scientific language is called being scooped, that you did something but somebody else beat you to publishing that same type of work, right? So Darwin was worried, and he shares Wallace's letter with his two friends, Lyell, who Wallace asked Darwin to share it with, and also Joseph Hooker. And the two men, and these two knew that Darwin had hit upon the idea decades ahead of Wallace. So as a result of this, the two men decide to they asked Darwin to quickly write a note of his own, and they forward these articles by these two men, Wallace and Darwin, to the annual meeting of the Linnean Society, which was to take place later in 1858. And in the note that they write, we can just read the first paragraph. This is a letter that was written by Lyell and Joseph Hooker to the chair of the Linnean Society, who was organizing that meeting. And they say that, my dear sir, the accompanying papers which we have the honor of communicating to the Linnean Society, which all relate to the same subjects, that is the laws which affect the production of varieties, races, and species, contain the result of investigations of two naturalists, Darwin and Wallace, and so on and so forth. Both men didn't attend the meeting that year because Wallace was obviously in Asia, and Darwin was mourning the loss of a child. His daughter died a little bit before the meeting was to take place, and he couldn't make it. So the papers were read by someone else for these two men in absentia, but interestingly, no one paid any attention to this. The society president later wrote in the meeting's summarizing minutes that nothing of significance was presented this year. This obviously, yes.
Why was Wallace in Asia?
Because he was still in.
And he was still working on that same thing.
Yeah. So this note was forwarded to the Linnean Society without knowledge of Wallace.
Okay.
Yeah, so he had just sent a note to Darwin asking him to forward it to Lil. And Darwin then shared with Lil and Hooker his worries about what was going to happen, that he was going to be denied credit. And Lil and Hooker knew that Darwin had been the first one to arrive at this. Right? How many of you have a class at 11?
Me.
All right. Okay. We will stop here. We still have a little bit more to do. So we'll take 15 minutes on Wednesday to talk about what happened next in the story. There's another extension that is really curious that happened after this. But let's close for now. We don't have time for questions. You can write to me or ask on Wednesday, but I will see you guys then. Okay? All right. Bye, everybody.
Thank you, sir.
Sir?
Below. Feel free to leave, those of you who have class. We're not going to do anything else.
Is there any group or something where I could get the links of the classes?
I will add you on Moodle, right? You will be on Moodle, so you will get the resources from there.
Okay, sir.
Yeah.
Still it is not registered yet. So I've just asked.
I'm sure it will happen. If it doesn't, just write. If it doesn't happen by Monday, just send me an email. I will add you manually to Moodle.
Okay, sir. Okay, sir. Thank you.
Okay.
So I know I may not be able to access the Moodle, but is there any way?
No, I think I will have to manually send you stuff, which I'm happy to do. No problems with that. So I will do it every weekend. After every class is not going to be possible.
Okay, thank you so much, sir.
Yeah, no problem. All right. No more questions? All right. Then have a good weekend.